Good morning, biology students. This is my first attempt at a YouTube video lecture, so hang with me. This is going to be on the first section of the chapter on DNA, or I should say the discovery of DNA. I also put in with your assignment a little video that I'd like you to watch that talks a little bit about the same thing we did here on the history of the discovery of DNA. So I guess what we start with here, we should go back all the way into the late 1800s with a guy named Friedrich Miescher, who's not on here. But basically, he gets credit for the discovery of nucleic acid. He basically took the gummy substance out of white blood cells, among other cells, but his main study was with white blood cells. And he found a gummy substance that was both acidic and it was from the nucleus of the cell. So that's how nuclein, his word for the potential genetic carrier, nuclein was his word for it. Later we called it nucleic acid because it was from the nucleus and it was in a slightly acidic substance. So Friedrich Miescher kind of took up where Gregor Mendel left off. There were a bunch of studies that we're going to go into a little bit later with DNA, but when we're looking specifically at the discovery of what it was that was carrying heredity, we probably should start in 1928 with the British scientist Frederick Griffith. He was trying to learn how certain types of bacteria caused pneumonia, and he did so in a famous experiment where he had two different kinds of pneumonia that were grown from mice. Okay, and basically he had two observations. There was one set of one of the disease-causing strains of bacteria had smooth edges around the cells when he grew them in culture. There's a little, like a little mucus layer around the outside of the cell. And the other one grew in colonies with rough edges. In other words, they didn't have this little mucusy, soft, smooth area on the outside edge. And what he did was he set up 4 individual experiments, and this is classic, controlled experiment. First of all, the mice were injected with disease-causing strain of bacteria. The mice developed pneumonia and they died. Okay. Secondly, the mice were injected with the harmless strain. They didn't get sick. They all lived. Okay. Next thing, Griffith heated the disease-causing bacteria. Now, keep in mind, he had an idea that there was something within the nucleus of these bacteria that was causing the hereditary trait of causing the disease pneumonia in one and not in another. He knew there was something in there that was carrying that heredity. So what he wanted to do was see what would happen if he injected mice with, let me back up. He heated the disease-causing bacteria, okay, which is basically killing them. But the material that caused it, the genetic material, was going to be in that mixture of material. He injected the heat-killed bacteria that were disease-causing into the mice, and they survived. The last thing he did is he took the mixture of heat-killed disease-causing bacteria with live harmless bacteria and injected the mixture into the mice, and amazingly they developed pneumonia and they died. Now obviously something from the heat-killed disease-causing bacteria got into the non-lethal form and transformed it heretically into disease-causing bacteria. So he concluded that the heat-killed bacteria passed their disease-causing ability on to the harmless strain. What was that material that did that? That's what we're trying to get at here. Now, Griffith called this process transformation because one strain of bacteria had changed permanently into another. In other words, the the non-lethal form was transformed into a lethal form by way of some material that was transformed into them. Griffith hypothesized that a factor must contain information that contain harmless bacteria into disease-causing ones. So remember that word factor, which was Mendel's actual term, or Merckmühl, and it would have been the actual genetic unit that was actually going to be causing this particular trait. So Fred Griffith got a lot of credit for his experiment. He was hypothesizing that it was the DNA, obviously, from the nucleus that was doing the transformation. Later on down the line, there were other experiments performed. The major experiments are the ones that get notoriety in the text. In 1944, we had a trio of three scientists, a guy by the name of Avery, McLeod, and McCarty. Three of them were a trio that wanted to take Griffith's experiment to the next level and exactly find out which of the molecules that were being injected were actually causing the transformation. Was it the gummy substance, the DNA on the inside, the nucleic acid, or was it a protein or a carbohydrate or lipid or even RNA molecules that were causing that transformation? So what they did was they kind of reenacted the Griffith experiment, made an extract from the heat-killed bacteria that they treated with enzymes. Now, enzymes, as you recall, are going to be substances that are going to break down or catalyze molecules. So he used enzymes that destroyed all the proteins, the lipids, the carbohydrates, and even the RNA, and the only thing that was left then was the DNA. Transformation occurred. So he knew that all these things, if all these things were broken down and weren't involved, the only thing left was DNA, and transformation occurred. So that gave them a good idea of what was causing the transformation. They repeated the experiment using enzymes that would actually break down the DNA, and guess what? When the DNA was destroyed, the transformation didn't occur. So therefore, they had the ability to make the solid conclusion that DNA was going to be the transforming factor that causes transformation. So we move on to a few other scientists, and we're going to hit the highlight just the people that made the news, so to speak. In 1952, there were a couple of scientists who took this one step further. Now, Avery and the other scientists discovered that the nucleic acid stores and transmits the genetic information from one generation to the next. What Hershey and Chase, Alfred Hershey and Martha Chase, were going to do is they were going to put a stamp on that. They wanted to make sure in another experiment to douse all the skeptics of this as to what exactly was causing heredity to be transferred from one cell to another. So what they did was they studied bacteriophages. Now, in order to understand this, you need to know how a bacteriophage works. It's a virus that affects only bacteria. And basically the way a virus works is that it will attach itself to a living cell. It won't survive without living hosts. It's kind of a parasite. When it attaches itself to a living cell, it will then inject its DNA or RNA, as the case may be, whatever the heredity material is for that particular virus. It will inject it into the cell, and it kind of holds up the cell. Okay? It holds a gun on the cell and says, all right, you do what I tell you to do, and that is make more of me. So basically, a bacteriophage is a virus that infects bacteria. It will inject its DNA into the bacteria. It makes the bacteria then start producing more of the virus until basically the cell is completely filled with new virus, and then the cell will lyse or break open, and it will allow all those new virus to escape and move on to new cells. So there are two kinds. There are the DNA variety and the RNA variety. And then there's also a protein code. So basically a bacteriophage is very simple. It's a virus that contains a round little covering over the nucleic material, which is protein, and it contains nucleic acid inside. In this case, it would be DNA. So when a bacteriophage enters the bacterium, the virus attaches to the surface of the cell and injects its genetic information into it, and the viral genes produce many new bacteriophages, which eventually destroy the bacterium, like we said. So when the cell splits open or lyses, hundreds of new bacteriophages will escape. So if Hershey and Chase could determine which part of the virus entered the cell, they could figure out what was the transforming factor. In other words, was DNA the hereditary material or was protein? And the way they did this was to use two radioactive markers. One of them was phosphorus, and one of them was sulfur. Now, we know now that, and of course they had a real good idea of what they were doing here, because they knew what DNA basically was about by then. They knew that DNA had phosphorus in it, but did not contain any sulfur. And they knew that proteins did basically contain sulfur. Many proteins contain sulfur. So they used these two markers to determine or follow which particular molecule entered the cell. So they grew viruses in culture dishes containing radioactive isotopes of phosphorus and sulfur. If S35 was found in the bacterium, they knew that it was the protein that had been injected into the bacteria and caused the bacterium to start producing new bacteriophage. If the phosphorus was found, the radioactive phosphorus was found, then they knew that it was the DNA that had been injected that caused the new bacteriophage to be produced. Nearly all of the radioactive material in the bacteria came from phosphorus. So therefore, that was conclusive proof that the genetic material of the bacteriophage was DNA and not protein. So we're getting closer and closer to the idea of what actually is the hereditary material in living things that causes traits to show themselves and to pass heredity on from one organism to another. Now basically, from this point, we want to move into the structure of DNA, which is section 2. But basically, we want to get an idea of what DNA's role is. Now, the role of DNA, as you probably would guess already, is three basic things. First of all, scientists wanted to know what was the actual unit that was storing this information and what was it made of. Now, Mendel called these factors, we call them genes today, but scientists were going to have to study the structure of that molecule to see how they were storing that information. And the second thing is they need to find out how they were copying that information and passing it on to new cells, which is one of the major parts of the next few sections we're going to talk about. And lastly, how do those genes express themselves? In other words, how does the structure of that molecule translate into an actual trait? like the shape of your ears or your nose or the color of your eyes. How does it actually express that? And that's what we're going to be looking at in the days and weeks to come. How does that happen?